All right, thank you. All right, thank you for the introduction, and if the program is still correct, I'm standing between you all and dinner, so I apologize for that. But bear with me for just a few more moments while I talk with you about the difference in response of the selected Bermuda grass cultivars to Bermuda grass stem maggot damage. So in the summer of 2010, Georgia producers began noticing this bronzing across their Bermuda grass hayfields. And if this photo was seen out of context, many would simply dismiss this as drought stress. I know I would. But when you actually zoom into the canopy, you can see that this chlorosis is restricted to those top few leaves of the tiller. And if you gently tug on these leaves, they'll easily slip away from the central shoot, indicating that this isn't drought stress, but rather damage caused by the Bermuda grass stem maggot, a new invasive species that is quickly spread from our South Georgia hayfields all the way north to Kentucky and as far west as Texas. As you may imagine with any new species, there's still a lot of questions to be answered, and there's very little literature available. So it should come as no surprise that control strategies, even today, are still very unclear. However, we know that cultural management, such as looking at resistant cultivars, is a major part of integrated pest management. And there was actually a study done in Japan that showed that suggested that damage was positively correlated with canopy density. However, we have much more information needed, such as looking at susceptibility of Bermuda grass cultivars that we use here in the Southeast today, and how the stem maggot damage impacts these. Also, to look at the effect of the damage on Bermuda grass yield and morphology. So for my master's research, I have three main objectives. First, I would like to compare the severity of damage among the selected cultivars. Second, quantify phenotypic variation in the cultivar's response to the Bermuda grass stem maggot damage. And third, characterize how the reproductive potential of the BSM differs with latitude in the Southeast. Due to my time restriction today, I'll only be focusing on these first two objectives. So in the greenhouse, I have 8 Synodon cultivars represented. 4 fine-textured Synodon dactylon, 2 very coarse-textured Synodon nymphoensis, and 2 relatively coarse-textured crosses between Synodon dactylon and Synodon nymphoensis. Fly treatments were imposed at 2 levels, with and without flies. The greenhouse bench was set up in a randomized complete block design with a total of 6 replications. Lastly, data was analyzed in SAS 9.1 using proc mix with rep set as our random effect. So just to quickly walk you through what a typical timeline would look like for one of my trial runs. After each harvest, I allowed the grass a 1-week rest period before I introduced my first pair of flies. Thereafter, every 3 to 4 days, another pair of flies was introduced for the remaining of the 4-week growing period before my next harvest. In 2012, I was able to conduct 4 runs, and in 2013, I was able to conduct 3. So at each harvest, we would clip the grass to a 5 centimeter stubble height, where we would then weigh the sample, very meticulously count the number of tillers, and determine if they were either damaged or undamaged. We would then evaluate morphological characteristics, including tiller diameter and leaf color that I'll present data for today, among others, which due to the time limitation will not be presented. So let's start by comparing the severity of damage among the selected Bermuda grass cultivars. We'll start by looking at the effect of cultivar on the number of infected tillers just when the flies are present, since it would always equal 0 when the flies are not present. So here on our y-axis, we have the average number of infected tillers per pot across our 8 cultivars here on the x-axis. And pulling across years, we see that those fine-textured Synodon dactylon tend to have a higher number of infected tillers per pot than those stargrasses or those with stargrass influence, with common Bermuda grass having the highest average number of infected tillers per pot. Yet many of you probably realize that not all of these Bermuda grasses have the same number of tillers. So putting this on a percent basis, our axis now here on the right, we see a trend begin to form, and this is similar to what the Japan study showed, and that those cultivars with a higher canopy density had a higher percent damage than those with the more open canopy. Still, producers are more interested in what is the yield impact. And so to answer this question, I looked at the average total dry weight in grams per pot across our two treatments. And since there was an absence of interactions between our treatment and year or treatment cultivar, or between the three, we can pull across those and we see a difference of about 1/4 of a gram between the two treatments. And this may not seem like a lot, but keep in mind this was a greenhouse study and it was enough to be significant. So let me put this into a more tangible number, because 1/4 of a gram, it's very small when you start thinking about haze usually harvested on acre, hectare basis. So it translates to a 7% decrease in yield. And if there are any Bermuda grass producers in the audience, as I imagine there are from the southeast, I know you want to argue that that's far too low of a number, and I agree. So I want you to keep in mind that this was a greenhouse study in an artificial environment where I got to control the fly population rather than the flies themselves. So just like those infomercials we all love on TV, your actual results may vary. Next, let's look at to quantify the phenotypic variation in the cultivar's response to the BSM. We'll start by looking at the effect of fly treatment on the number of tillers per pot. What we see when we look at the average number of tillers per pot, again, just across our two treatments due to the absence of the interactions, we see a difference of about 6 tillers, which again doesn't seem like a lot, but that translates to an 11% decrease in tillers. And since that is a yield component, you would expect the decrease in yield that we saw in the earlier slide. And what literature suggests is that as the number of tillers decreases, the weight per tiller should have increased. So keep that in mind for just a moment when we get to the later slides. Next, let's look at comparing the fly treatment effect on the tiller diameter and leaf color. Let's start first with tiller diameter, here on the left-hand side on our y-axis, across our two treatments. And we see that in the presence of the fly, the tiller diameter actually significantly increased. Likewise, with leaf color, we see that in the presence of the fly, the leaf color tended to darken, our axis now here on the right-hand side. At this point, with the information we have available, we want to attribute this to a reallocation of nutrients and photosynthase within the stem once those top leaves are damaged. So I know this anticipation's been killing you, but let's look finally at the effect of the fly treatment on the weight per tiller. So we have our average weight per tiller here on our y-axis across our two treatments. And as I suggested earlier, there is not actually a significant difference between our treatments in the weight per tiller. So while the Bermuda grass stem maggot is creating conditions that should have been conducive to increasing the weight per tiller, the stress is simply too much for the tiller to take advantage of and actually increase weight, suggesting why we saw a yield decrease in those earlier slides. So preliminary conclusions are that the percent of tillers damaged and the severity of damage depends on the cultivar. The presence of the fly increases tiller diameter, darkens leaf color, decreases the number of tillers, but does not affect the weight per tiller. And while I didn't have the opportunity to present the data, we did see that reproductive potential did vary with region. Some implications that we've drawn, or we predict, are that the laying groundwork for future research. Most importantly, assessing the economic threshold using a fine cultivar such as common or coastal Bermuda grass with a coarse cultivar such as Tipton 85 as a more tolerant control. Also, we would like to evaluate factors driving the regional difference between those populations in terms of reproductive potential, specifically first looking at forage management and how this could have impacted those population differences that we saw. Lastly, we can suggest more guidance for producers. You heard in the presentations earlier today about all those keys for increasing your forage yield. And so one of the ways that we can do that is if producers live in an area that has been historically prone to the stem maggot damage, is just starting with a more tolerant cultivar, such as Tifton 85 or Coast Cross 2 if it's released to the public. Some future directions we'd like to take is understanding the life cycle of the species, because at this point we know very little. Quantifying the economic impact of the damage, and proposing appropriate and economical suppression efforts. So at this, I'll take any questions before I fly out of here. Yes?
No question. I don't want to about these flies, but when you introduce the flies, is there like a net or something over that particular pot to ensure the flies were impacting that pot?
Right. So the question was, how do I ensure the flies just, I guess, damage the specific experimental unit I want it to? So I actually designed a acetate kind of plastic and mesh structure because, as you may imagine, there's nothing readily available in the market for keeping flies on a Bermuda grass pot. And so lots of gluing and taping later. It's actually about a 3-foot cylinder that will fit on a 6-inch Bermuda grass pot. And to introduce the flies, I catch them in the field in a sweep net, bring them back to the lab, separate male and female, put one of each in a pot, and I just lift up, the acetate mesh structure. I can open the vial and release the flies, and they actually fly towards light, and so we take advantage of that and have lights on the greenhouse bench, and they'll fly straight up.
Where is this fly native to, and how long do you think it's been in the United States?
So the question was, where is the fly native to, and how long has it been in the United States? Is that correct? All right, so the fly is thought to be native to the Southeast Asia, kind of oriental region. It was first found in a Swedish-Chinese expedition. There's very limited literature available on this because there was other pests that were causing more damage in that area, and this one just happened to be there in many cases. The first sighting of the fly confirmed in the continental U.S. was in 2009 out in California, but as we said, the first known instance of the fly in Georgia wasn't until 2010, and we're not sure how far before that it could have been here to build up a population that we would have seen. It was likely that Georgia was in a drought in that time period, and if you don't know the difference, you would have just dismissed this as drought stress. So we could have thought it was drought stress for years and not understanding until the fly was caught that it was actually a pest.
We've noticed more damage in plots that have received more nitrogen fertilizer. Have you seen that too?
So the question was, do I notice more damage in plots that receive more nitrogen fertilizer? Yes, we do. In our research plots, most definitely receive more damage in those control areas around the plots. And I notice in the hay fields that I sweep, if a producer takes better care of their hay field and that they irrigate it and fertilize it and manage it properly, there's always more damage than their neighbor that just kind of lets their hay field or pasture go.
There's a session tomorrow, by the way, on covering more of the details of the history of what we know about these insects so far.
So.
The idea to fix the plant or fix the fly? Get rid of the fly.
So the question is the plan to fix the plant or fix the fly? And if you've ever tried to deal with these flies, they're very pesky, and I don't think we'll ever be able to fully control the fly, and more of that will be addressed tomorrow, I believe, in terms of control strategies that you can take in terms of certain things you can spray and mechanical suppression. But there's been research that has tried to eliminate similar other Anthragona species, and they've proved unsuccessful. They can reduce the population, but they can't eliminate it just because of the fly lives inside, or the larva lives inside the stem, and there's not currently insecticides that can work systemically that are approved for hay fields. Changing the grass would be, I think, easier just because we have those available, such as Tifton 68 and Tifton 85, that prove to be more tolerant. They're not totally resistant, but they're certainly more tolerant. And Coast Cross 2 showed, just tremendous tolerance in the greenhouse. So I hope that one's released to the public so producers can take advantage of that forage.
Any other questions?
When you were sweeping, did you detect any specific ratio of males, females?
So the question was, when I was sweeping, did I notice a specific ratio of males to females? It depended on the region that I was in and also the time of year. Up closer to Athens in central Georgia, we tend to notice a higher population of females, or a ratio of females to male. Down near Tifton, it's almost always 3 females to 1 male. And Dr. Will Hudson has more data on that than I do because he's been working on this problem longer. So his records, spread over about 4 years, where mine are a year and a half that I've been in Georgia.
Please, one question. Any screening for fungal diseases that might be interacting with the plant?
Right. So the question was are there any fungal or screenings for fungal diseases that could be interacting with the pest and the plant?
Yeah. In Florida, we've been tracking the same issue, but we are more prone to diseases.
Mm-hmm.
And in some cases, we find no evidence of the fly, but we do find take all root rot with the exact same
Right.
symptoms.
Right, so same question. This may be in Dr. Hancock's video presentation tomorrow, but we have noticed in Georgia that there are some cases where the leaf spot is often in the same fields that are damaged by the Bermuda grass stem maggot. And so as you saw with my data where the weight per stem should have increased, it's obvious that there's some physiological damage going on that we don't yet fully understand that could make the grass more prone to other, the fungal diseases, the root rots, etc.